Hello, I'm currently in a molecular neuroscience course here at UC Irvine, and I'm having to give a presentation on this paper. It's a classic, I guess, in the field now. It's almost a decade old, but it's still has great lessons for us in neuroscience today, I think. And so, yeah, just thought I'd share this presentation with you. And this paper is called The Precise Temporal Pattern of Prehearing Spontaneous Activity is Necessary for Tonotopic Map Refinement. Okay, and this is out of the Candler lab. And let's dive into the background material. So here we have, this isn't the auditory pathway we'll focus on, but this is a very common theme for an overprojection of axons from the sensory system into deeper in the brain. And it's this diffuse initially topography that's sharpened up through spontaneous activity and once the animal is born and sufficient development occurs that sensory signals can be transduced, right? So hearing starts or vision starts when you open your eyes. So this is projections from the retina to the tectal area, and you can see with experience that it's refined and you have a sharper retinal topography represented in the tectum. So this doesn't happen just for the visual system. This also happens for the auditory system. And so you can see here this inner supporting cell on the left, which is part of this transient structure that goes away during development, the Kolliker's organ. It releases ATP, which binds onto the inner hair cell. Okay, that's going to let a bunch of potassium in, which depolarizes the inner hair cell, causing it to release glutamate onto this spiral ganglion neuron, okay? And this will be faithfully propagated through the rest of the auditory system from the inner ear, where we are right now in the cochlea, all the way into essentially the primary auditory cortex. We won't go that far. We'll mainly just stick around in the brainstem near the pons for most of this. Okay, and what they do in this paper mainly is they realize, these acetylcholine receptors, there's specific ones with subunits made of alpha 9 and alpha 10 acetylcholine receptors. These are nicotinic, so they're fast reacting. They're weird in the inner hair cells. They're only expressed there, and they are closed normally. Normally acetylcholine receptors are open and then closed when binding to acetylcholine. But in here on the inner hair cells and the outer hair cells, it's actually the opposite, it seems. They will actually they will be opened by acetylcholine letting calcium in. And this is this is shortly washed out by these potassiums, which are nearby, are triggered by this calcium influx, and so the potassium will rush out of the cell. And so this whole thing, this feedback from the brain stem, this MOC pathway that dumps acetylcholine, is actually inhibitory. So that's what makes these doubly weird, is these subunits are only expressed in the hair cells of the cochlea, these alpha 9, alpha 10 acetylcholine receptor subunits, and they're inhibitory. And so that's what they actually did with these knockout mice, is they just knocked out this acetylcholine receptors, and it shouldn't affect the rest of the brain, just these hair cells and this feedback pathway from the brainstem. Oh, and you can see one other minor difference, right, is this feedback occurs on the outer hair cells, like shown here, but it's actually on the spiral ganglion dendrites for the inner hair cells, but it's essentially very, very similar, right? The inner hair cell could keep dumping glutamate all at once, but if you're getting feedback from acetylcholine from the MOC in the pons, you can shut down the spiral ganglion all day. Okay, and these are going to be the circuits we're looking at. We've got the cochlea, where we had those inner hair cells we were looking at. The ventral cochlear nerve, which has tonotopy, just like the cochlea. In this upper diagram, the shading, the white color shading, is the high frequency information, and the darker shading is the lower frequency. So this is the tonotopy. You have the B down here and the B up here. Okay, and we can show you have ipsilateral connections from this cochlear nerve to the MNTB near this pons medulla space in the brainstem, and you have ipsilateral connections with the LSO and the ventrocochlear nerve, right? So this one crosses over and this one doesn't. And these are both excitatory from the spiral ganglion cells, or actually this isn't spiral ganglion, sorry, but from the cochlear nerve axons are excitatory. And this MNTB pathway to the LSO is inhibitory. And this is mainly what we're going to be really looking at with the majority of this review paper. When reviewing this paper, we're going to be looking at this inhibitory map. And they're both tonotopic, and the projections from one area goes to the other area, right? You see this. The dark goes to the dark, and the white light goes to the white light. And this gets screwed up when you knock out the alpha 9 receptors. And this figure down here is showing very much the same information. It's a little more colorful. But notice this LSO, the SO stands for superior olive. It's this kind of whole bunch of nuclei in this area around here. We've got a MSO, which is some other part of the superior olive, and not shown here is the MOC. And that is also part of the superior olive in this brainstem, pons region. And this would project back to your inner hair cells, right? The MOC is what we saw earlier right here. So we don't really go any higher up, I believe, in this whole paper than just the brainstem and all of these nuclei talking to each other. Okay, so we've done sufficient amount of background to actually jump into the results of this paper. So they start recording from those MNTB, right, that inhibitory region projecting to the lateral superior olive, the LSO. And in the wild type mice, you have the spontaneous activity. This is pre-hearing, right? So this is not the mice are born and they're hearing anything yet, but they're born for a while, and the cochlear nerve actually doesn't transduce sound until about postnatal day 12 or P12. And so what we see here is we see these blue lines in the wild type, the normal mice, are showing bursts of activity. This is all spontaneous. There's no hearing transduction. This is just like waves of activity that are normal in development and help set everything up. And we have this little red bracket that's showing where we're going to zoom into when we go over here. So you see we've kind of zoomed in. We've got a little bit of this noise is easier to see. And inside individual bursts, we've got, even kind of stronger, shorter little pairs of individual action potentials. So we've got, these look to be, I guess, groups of 2 action potentials when you zoom in here. And this is it's a big cell, it's a big synapse driving this, so you can see the influence. They call this the presynaptic. And then afterwards you see the consequence from recording this MNTB neuron. You see the postsynaptic. So this is actually the action potential of the cell you're seeing, the postsynaptic component. Okay, and so it's kind of similar when you knock out the alpha 9 subunits in the MNTB when recording individual neurons, but you'll notice that the burst duration is a lot shorter. But if you zoom into an individual burst, this is the same duration we're zooming in on, you actually have higher frequency bursting. So less bursting, but once you zoom in, man, we are going crazy. But if you zoom in enough again, these individual spikes looks very similar. The noise looks very similar. We're kind of changing not the overall activity levels, but instead we're changing burst dynamics. And we're going to look at essentially the same information, just quantified in histograms. You need to know what an ISI is. It's an inner spike interval. So this is an action potential here and an action potential here. The ISI is just the duration, the time duration between these subsequent spikes. Okay, and so what this is showing is with the wild type, with the normal mouse, you kind of have this bimodal distribution. You either have to wait a long time between spikes, or you kind of have more burstiness, right? Yeah, this 10 to the 0, so it'd be like 1 millisecond around here, which is shorter than the refractory period, right? So this just shows kind of bursting and then shorter individual spikes probably over here. You have more of a unimodal when you knock it out. You don't have bursting and individual spikes. You just kind of have everything kind of grouped together. And a lot more burstiness is represented. You can normalize these two so that you can compare them more faithfully. And yeah, you see this middle region really is overrepresented. So the takeaway is the overall level of activity really isn't changed much, it seems, just looking at it, but there are subtle spike timing characteristics and burst characteristics being altered here by the knockout. Okay, this is a continuation, kind of what I said, right? Is the overall firing rate is very similar. The bursts per minute are similar, but the burst duration is much shorter in the knockout, but this is compensated to get to the an equal overall activity level by increasing the burst frequency within bursts. Just how fast you're firing within bursts is increased in knockout. And then they also looked at individual loner kind of spikes are higher, and they looked inside a burst and classified these mini bursts, and they go, well, we have more higher frequency of mini bursts as well. Okay, now what we're going to actually be doing is uncaging glutamate onto the MNTBs. So this is that region, right? The MNTB region that sends an inhibitory projection to the LSO. So we're going to be exciting the MNTB, causing inhibitory axons to fire in the LSO. And so we want to record from individual LSO neurons while we probe the MNTB to see which parts of it influence the downstream LSO neuron. And so you see here, right, at postnatal day 3 in the normal wild type mice, this whole region can be stimulated, excited in the MNTB, the upstream region, to elicit activity in the downstream individual LSO neuron. Okay? But as development happens, so this is P12 is about when hearing onset is. So this is all like mainly due from spontaneous activity and natural cues. You see the sharpening effect. You can't stimulate LSO neurons as easily. You have to be more specific. Your area of MNTB that you can uncage glutamate on with a laser to drive activity in the LSO neurons, or well, rather not drive activity, but rather inhibit activity, right? This area is getting smaller. So there's probably some structural and functional changes underlying this, and we'll get into that. But in the knockout mice, the area actually stays the same. So already developmentally in the spontaneous periods, pre-hearing onset, we've got something funky going on. This narrowing, this focusing of the tonotopy is not occurring. These graphs show essentially the same thing. This one's area. You see the area doesn't change on the alpha knockout mice like it should in the wild type. So we have a higher area of influence in the MNTB onto LSO neurons. And really this is area, but this is just width, right? The width is the tonotopy. So the height is very similar, so they just focus on the width and you see very much the same trends. So this is showing, right, this is a nice little sanity check. It's nothing to do with the other dimension that we don't care about. It's all across the tonotopy that's developing weird. And this is maybe the hardest to experiment to understand, and then we'll get into some stuff that we can probably pretty much fly through. So they stick an electrode in the MNTB axons here, these nerve fibers, and they stimulate it. They drive current in, and they'll use different amounts. And then they're just recording again in the LSO. We were recording in the LSO last time, just individual neurons, right? So you latch onto a neuron and you start stimulating. You just start turning up the stimulation until you get the smallest response. You get a little bit of deflection, and they assume that, notice all this noise, it's because we're zoomed into picoamps. That's like no current at all. But there's a tiny deflection. So the idea here is that we're recording the influence of a single neuron in the MNTB to silence the LSO. So we're hyperpolarizing the LSO, so some amount of inhibition has been generated, right? And we note when this minimum amount of driving current is to trigger to trigger this small first deflection we see, and we think we're capturing one axon. And then we'll do very similar. We'll turn it up, and we'll just keep turning it up until it doesn't change this anymore. Notice we've gone up like 3 orders of magnitude to like a 0.2 of a nanoamp. We're looking for the saturation. Once we turn up the driving current such that it just doesn't change anymore, we'll also mark that as the maximum current. And that's the idea there, is we're activating all of the inhibition that can be driven onto this individual LSO. So we're kind of looking at the minimum influence from one axon and the maximum amount of inhibition from all the axons going to this one LSO neuron downstream. Okay, and they're very similar at P1 and 3, but the difference, let's see, the black one is the wild type. So the wild type actually has a much stronger minimum influence. So just one axon from MNTB is much stronger inhibition in the wild type mice. But in the knockout mice, these stay weak. They increase a little bit, right, developmentally, but they're much weaker in individual MNTB neuron is much weaker at inhibiting an LSO neuron. So that's okay. We've got a great target for something getting screwed up by this knockout. But something really interesting is the max doesn't matter. So this hints at like kind of a homeostatic set point. There's somehow an ability to balance this. Even though the individual inhibition is goofy, the overall level of inhibition for this LSO area seems to be able to compensate for this knockout effect. And a convergence is just a ratio of the average max to average min current. And we see that the convergence is normal in the beginning again, but with development, the alpha 9 knockout has much higher convergence, almost double by average. So this points to maybe like double the amount of inhibitory synapses going from MNTB neurons to an LSO neuron. So even though there's weaker single projections, you double up those amount of projections relative to the wild type, and thus you get a similar amount of inhibition. And G is essentially showing the same thing. These cumulative probability graphs always trip me up, so I'll just go ahead and skip that. What this is showing is the decay and rise times. And you see on the right, they're the same developmentally. The decay time decreases, as does the 20 to 80% rise time characteristics. But really what they're showing is they took a first order linear or first order exponential fit model of these currents entering and exiting the cell throughout different conditions, right? P2 versus P10 and wild type versus knockout. And they're very, very, very similar in these individual spike settings. And what they're trying to show here is that the neurotransmitters didn't change, the distance didn't really change from the synaptic connections, the release probability of vesicles didn't change. This is just a sanity check that developmentally these individual spikes look very similar in their dynamics, how the onset and the return to baseline occurs. Okay, and we can reconstruct, we can label axons, right? So we've been looking at these, we've kind of been looking at electrophysiologically the influence of an MNTB axon and an LSO neuron. We can capture those in those axons by biopsytin. And then we can look at them under a microscope, take slices of them, and reconstruct them with computer aid. And so that's kind of what we've done here. And so we've labeled them at P4 in the wild type mouse and then P13. So this is very much capturing pre-hearing onset development. And we see that LSO in the blue outline, the blue blob, is growing a lot during this week, but so are the amount of axonal coverage and projection, synaptic connections of that axon. And it's very similar in pre-hearing in the knockout mice as well, right? We see the LSO is growing, and we see the amount of connections and the coverage of LSO per MNTB neuron also increases. So very, very normal. Right, and I've said this at the bottom as well, right? Pre-hearing onset axon development is normal in the knockout mice. So these all look very similar. We're looking at the number of connections from the axon to LSO neuron, so the Bouton number. We're looking at the Bouton area and the Bouton spread, right, which is a subset of Bouton area. They all look developmentally normal. Same type of information. We're just drawing heat maps of where these Boutons are, where these synaptic connections are made. And you see that going from day 2 to day 12 in the normal mouse on the left, the wild type mouse, there's a lot of density added at the middle. right? You see how it gets red in the middle. And if we look at the knockout mice on the right, we see the very, very same trend. There's some spread, but most of the addition of axons are towards the middle of what was already established at near birth at P2, P4. And this shows that synaptic growth, synaptogenesis, is normal before hearing onset. And this is just a histogram of the same results. The relative Bouton position is on the x-axis, so 0 means the center of that density. And you see that we are growing. P2 is below here in gray, and we add a bunch of synaptic connections to the axon. So we go up, and we see the very similar trajectory in the final destination in the knockout mice. Okay, so things have been looking kind of decent. So what's going on with that lack of sharpening? When are we going to explain this? And this is our first kind of hint at what's going on. Is there's actually atypical axon pruning. We were just looking at, like, we just stopped looking at P12, but if we continue on about another week and we look at P20, this is where things start to go awry. If you look at the wild type mice going to P20, it gets a lot smaller, and not in the height, but into just the width dimension. And this is the dimension again of the tonotopy. So this is maybe explaining why normally, developmentally, you see a sharpening of MNTB being able to drive activity in LSO, is because the area of influence, the number of neurons it's connected to, is decreased. A sharper, thinner width of influence. But we totally lacking in the knockout mice. This sharpness, the sharpening of this band just does not occur. And we see the same thing when we look at it quantitatively like this, right? The number of connections stays high, the area stays high because the width of that tonotopic map stays high. Okay, we can look at the heat maps again, right? And something interesting is earlier we were looking at these heat maps, we were adding connections, now we're actually removing connections. And really, it's kind of like a sinking iceberg. The connections are uniformly kind of pruned. It's nothing to do with inappropriate connections. It seems. It just seems that if all of these have equal probability of being removed, and so you just kind of have this sinking effect. You can shift the top one up here just down, and it's got the same sort of curve. And yet again, right? This axon pruning from onset of hearing P12 to P20ish and knockout just doesn't occur at all. They're almost overlying each other, the synaptic densities in the LSO. And so just to show that this isn't something different about the spontaneous activity post-hearing or the environment distribution of noises coming in is weird. They did a bunch of white noise rearing mice experiments, right? So they've shown this in the past where you take mice and early on in development, like starting like P8, so before hearing onset, you just put in a bunch of white noise, which has has equal distribution of all spectral frequencies. So all different kind of notes are in there. And you can pulse this randomly, like 30 times a minute for about a second each. And this really messes up the tonotopy in the cortical areas, at least I know of. But they did this in the mouse, and there wasn't this atypical axonal pruning in the white noise reared mice. So they know it's not something to do with post-hearing spectral distribution of input, right? It's not what they're hearing probably the spontaneous activity after hearing that's messing with this, but instead it's something earlier in development. And so they did some more sanity checks, and they labeled they put in some biocytin into the ventral cochlear nuclear on one side, and just looked and made sure that there are no inappropriate axons. So inappropriate axons would be in these areas here, and there's really none here. They could find some axons labeled, but they weren't synaptically coupled to each other. So that's good. So this was just a sanity check that the projections aren't messed up. And in addition, they looked at the synaptic connections from VCN to MNTB, and they're these big calyx. I think this is the calyx of Held, or the end bulb, one of those. But they're both giant glutamatergic synapses that synapse onto like one neuron. In this case, we're looking at an MNTB neuron. And this is at postnatal day 14, so just a little bit after hearing onset. And I mean, if I didn't tell you which of these were which, I don't think you'd be able to discern. So they look very normal as well. And so let's just go over the discussion now that we've got all these results. I was thinking this, and they brought this up in the discussion of the paper. I was so happy I was on board. I was asking the right questions. I wanted to know, well, what is causing, or how is this knockout causing changes in spontaneous activity in the MNTB? If we know this knockout of these alpha-9 nicotinic acetylcholine receptors is only occurring in hair cells, how are we influencing like 4 synapses away the MNTB spontaneous activity? And it could just be, like we said, you're getting rid of an inhibition. So especially on the inner hair cells during development, this MOC is developing as well. So it's not like it's always like this. This is after development. So sometimes the inner hair cells are the only ones being innervated. Sometimes it's probably by volume transmission. They're not directly connected. And so it's changing a lot, but it's thought that maybe this tonic inhibition from the acetylcholine is really important, right? And this makes sense of, we saw higher runaway bursts that didn't last as long. It could be because they're running out of steam faster because there's not an inhibitory acetylcholine projection. It could also be that the vesicle formation in the inner hair cells and the outer hair cells are goofed up. It would be more important to look at the inner hair cells, as these are the majority of the efferent projections from the inner cochlea. Another takeaway from this is noting, right, that the total inhibition in the LSO is homeostatic. It's like this heater on the left. They just set it to 72 units of inhibition, and no matter what the single axon contributions are, we're going to end up with this amount of inhibition. Okay, but the single axon contribution is variable. So we saw that we ended up with a lot weaker, higher number of synaptic connections between the MNTB and the LSO. And so maybe part of this is the idea of the authors is that the timing is what's really matters here. Is even though we saw subtle timing differences in spontaneous activity, that the timing really matters for the weight changes. And so you can see here's something like STDP learning curves for inhibitory neurons have been suggested before. So even if you don't understand what this is showing, it's showing that the spike timing really matters if you're going to make the connection stronger or weaker. And, last but not least, there are distinct periods of functional, so kind of strength, and structural refinement. The functional refinement is pre-hearing onset, right? We saw this in the wild type mice. We saw the sharpening right before hearing onset of the influence of NTB onto LSO. And we saw equally in the electrophysiological recordings when we were driving current in there, that those individual influences got a lot bigger over time as well, right? In the wild type mice, just developmentally from P2 to P11, your single smallest influence you can have downstream gets a lot bigger. So that's all pre-hearing, but once you're post-hearing, those functional refinements quickly get changed into structural refinements in the normal mouse, right? You have that iceberg sinking, and that iceberg sinking just does not happen with the knockout mice. Okay, and finally, in conclusion, eliminating feedback inhibition at the hair cells by knocking out alpha-9 nicotinic acetylcholine receptors causes subtle timing changes in spontaneous MNTB activity without changing the overall levels of activity. KO mice have much less developmental sharpening of their inhibitory tonotopic map. This involves atypical connection strengthening and an absence of axonal pruning. And last but not least, precise spike timing characteristics in the MNTB before hearing onset is very important for establishing sharp tonotopy in the LSO. Okay, and that is my presentation. Thank you guys so much.